This presentation is in response to exam 2C, question number 2, comparing and contrasting MHC types. The MHC, or the major histocompatibility complex, is a set of cell surface proteins essential for the acquired immune system to recognize foreign molecules in vertebrates, which in turn determines histocompatibility. The main function of major histocompatibility complex molecules is to bind to antigens, which are derived from pathogens, and display them on the cell surface for recognition by the appropriate T cells. MHC molecules mediate interactions of leukocytes, also called like blood cells, with other leukocytes or with body cells. The MHC determines compatibility of donors for organ transplant as well as one's susceptibility to an autoimmune disease. There are two major classes of MHC molecules, both of which consist of an alpha and a beta chain, but they are from different sources. MHC class 1 molecules consist of one membrane-spanning alpha chain, a heavy chain, produced by MHC genes, and one beta chain, or a light chain, produced by the beta-2 microglobulin gene. MHC class 2 molecules consist of two membrane-spanning chains, alpha and beta, of similar size, and both produced by MHC genes. In each case, the MHC molecule has a groove that binds a peptide, which it can then present at the cell surface to a T cell in order to elicit an immune response, because T cells only recognize antigens as complexes with MHC molecules. The two classes of major histocompatibility complex proteins differ not only in their structure, but more importantly in their functional roles within the immune system. The two types of MHC molecules are specialized to present different types of antigen and thereby elicit different types of responses. So with the class 1 molecules, MHC1 glycoproteins are present on almost every cell in the body, acting to present endogenous antigens that originate from the cytoplasm. These antigens include not just self proteins, but also foreign proteins produced within the cell, such as viral proteins that take over the cell's machinery in order to replicate the virus. When these proteins become degraded, the peptide fragments can be transported to the endoplasmic reticulum, where they can bind to MHC1 proteins before being transported via the Golgi apparatus to the cell surface. Once at the cell surface, the membrane-bound MHC1 protein displays the antigen for recognition by special immune cells, these cytotoxic T cell lymphocytes. MHC1 proteins work to present the types of proteins being synthesized within a cell, which can then be monitored by killer T cells as part of a surveillance system that identifies and destroys any cell with overabundant or unfamiliar peptide antigens. These would be malignant cells or cells harboring viruses. MHC class 2 glycoproteins are only present on specialized antigen-presenting immune cells, including macrophages, which engulf foreign particles, dendritic cells, which present antigen to T cells, and B cells, which produce the antibodies. MHC2 proteins present exogenous antigens, unlike the endogenous antigens presented by MHC1. These exogenous antigens originate extracellularly from foreign bodies, such as bacteria. Upon encountering a pathogenic organism, proteins from the pathogen can be degraded into peptide fragments by the antigen-presenting cell, which then sequesters these fragments into the endosome so they can bind to MHC2 proteins before, of course, being transported to the cell surface. Once these are at the cell surface, the membrane-bound MHC2 protein displays the antigen for recognition by a different type of T cell, namely the helper T cell lymphocyte. These helper T cells are activated upon binding to macrophage or dendritic cell MHC2 antigen, causing the release of lymphokines that attract other cells to the area of infection in an attempt to confine and destroy the antigenic material. In addition, the binding of helper T cells to B cell MHC2 antigen stimulates the development of a clone of antibody producing cells against the antigenic material. So the major differences between the two classes: class 1 is comprised of an alpha chain and a beta 2 microglobulin chain, whereas class 2 is encoded just alpha and beta chains, though both are encoded by the MHC genes. Class 1 is present on most cells, whereas class 2 is present only on the antigen-presenting cells. Class 1 binds to endogenous antigens. Class 2 binds to exogenous antigens. Class 1 presents antigens to cytotoxic T cells. Class 2 presents antigens to helper T cells. Class 1 binds CD8 adhesion molecules on its target cells, the cytotoxic T cells. Class 2 binds CD4 adhesion molecules on the helper T cells. So MHC class 1 is stimulated by the presence of foreign or overabundant antigens and targets cells for destruction. Whereas with the MHC class 2 molecules, the presence of foreign antigens induces antibody production and attracts immune cells to the area of infection.